I get to start by asking all of you a question. It seems to go without saying right now in our lives to understand that life comes from other life. In fact, you've probably known that since you were a tiny little person and you understood that little babies come from mommies and daddies, right? That may have been something that was inherent in your very young learning. However, this hasn't always been straightforward. And so my question is, how many years ago did scientists accept / fully support that all life arises from other living things of the same kind? So we want to know, was that, like 1,500 years ago? Was it 227, 139, 457? I mean, how long ago was it that we finally got it, that life comes from other life? So I'm not going to answer the question right now. I want you to be on the edge of your seat waiting for an answer as we dive in to the first of our historical coverage on microbiology. The Earth is thought to be some 4.5 to 4.6 billion years old, and of course it's been of great debate as to when life arose on the planet. However, in 1977 there was a beautiful fossil of an Archaean chert that was dated to about 3.5 billion years ago. So that it's thought that life has existed on Earth for some 3.5 to 3.8 billion years. And of course there's been much debate on the origins of that life, right? I just said life comes from other life. So the question is, what about that first abiotic event, right? That is, what did life come from to start with? And there's a man named Walter Gilbert who has put forward a hypothesis called the Rna Hypothesis. Now, this is very much based in the work of many scientists, including a man called Thomas Cech, who's at the University of Colorado. And this is the idea that RNA predated cellular structures. That is, RNA was the first macromolecule to exist that could act as both the carrier of genetic information, right? We know that RNA has nucleotides of which it is comprised, and that it can serve as a genetic code, but that it also could act as a catalyst. And that's something that we have only more recently realized, is that RNAs can have enzymatic activity. We call them ribozymes when they act as enzymes. So the idea that life evolved from RNA is a very poignant one. It's crazy to think about life having existed on Earth for 3.5 to 3.8 billion years ago, and we really didn't understand that microorganisms were out there for a majority of the history of humanity. That is, it wasn't until 1673 that a man named Antony van Leeuwenhoek, for the first time, very astutely described microbial life. He did so by making a microscope like the one that you see in this figure that can magnify some 50 to 300 times, enabling him to see life in everything ranging from a drop of pond water to his own urine. He liked to look at everything under the microscope. And let me tell you, it was his labor of love. He was not a microscopist by trade. Instead, he was a draper and a haberdasher, meaning that he made men's clothing. He lived in the Netherlands, and he occupied his unused hours by making lenses. And his lenses were incredible. They had higher magnifying power than the lenses that Hooke and Galileo made. So he was able for the first time to really give these epic descriptions of the microbial world. And when he saw these microorganisms, he called them little animals, or animalcules, and he wrote these animated letters to the Royal Society of London. I'm going to read you one of these letters, an excerpt. Very many little animalcules, whereof some were roundish, while others a bit bigger consisted of an oval. On these last I saw 2 legs near the head and 2 little fins at the hindmost end of the body. Others were somewhat longer than an oval, and these were very slow a moving and few in number. These animalcules had diverse colors, some being whitish and transparent, others with green and very glittering little scales, others again were green in the middle and before and behind white, others yet were ashen gray. And the motion of most of these animalcules in the water was so swift and so various, upwards, downwards, and roundabout, that it was wonderful to see. Imagine his excitement when he was seeing them describing the microbial world for the first time and sending these letters off to the Royal Society. I mean, we get jazzed when we see these microorganisms for the first time under our microscope. And guess what? When you guys come to lab on February 3rd, we're going to be checking these out, and hopefully you'll see some things like amoeba on the upper left here, and a ciliaphoria here on the lower right. You can see the movement of the cilia. We're going to check out some of those characters that are often found in pond water and the protease that we see there. So let's use Antony van Leeuwenhoek as sort of the birth of microbiology, even though we know that it's always been there, and there were people that were speculating to the existence of microorganisms prior to Antony van Leeuwenhoek, and rarely is the person to actually first do something ever credited. It just happened that he did a really good job of describing it and sending it off to someone who could record it. And so we say, here's the birth of microbiology, and how would we define microbiology? Well, this is tricky. It used to be that we said that microbiology was the study of life forms, but that's not entirely true, because we study things like viruses and prions in microbiology, and they're considered to be unliving, acellular agents. So now we often define microbiology as life forms and agents that are too small to be seen with the unaided eye. But that definition even falls short sometimes, because occasionally we study microorganisms that aren't really too small to be seen with the unaided eye. In fact, there are even some bacteria that are the size of a period on your paper, and they can actually be seen without a microscope. So knowing that there are exceptions to every definition, I want to talk just a little more about the difference between life forms and agents, and to kind of give ourselves a starting point for understanding life forms and agents, we're going to go ahead and take a look at some of each. Now, life forms are cellular in nature, and in fact, we might even just put that in parentheses here, describing our life forms as being cellular versus our agents as being acellular. In fact, we can see the cellular structure of the life forms, which could include things like this bacterium. The little hair-like structure on this microscopic field is actually termed E. coli, Escherichia coli. It is a bacterium. So these are bacteria, right, the plural of bacterium. These are yeast. So yeast are eukaryotes. Bacteria are considered quote-unquote prokaryotes. We'll talk more about that term. Yeast, and over here, this is a volvox, which is actually a protist. Now, not pictured here is another cellular form of life called the Archaea, and I mentioned that it was an Archean that was found in Australia that was dated to 3.5 billion years ago. So Archaea are often typified as being very ancient. So the Archaea, the bacteria, and the eukarya are considered life forms, whereas agents will be things like viruses. You might recognize this epic virus as being HIV. This is a prion, so remember mad cow disease and CWD. These are diseases caused by proteinaceous infectious agents. This is a protein, a prion. This is actually a viroid. Viroids are infectious agents of plants. It's comprised only of RNA. There are also something called satellites that are made up of RNA or DNA. So agents, as you can see, are not cells, whereas life forms are, and there are different categories of each. And you can look forward to talking about these more as we go throughout the rest of the semester. For now, let's leave this behind so that we can dive back into the history of microbiology, and I want to talk about the process by which scientists had to disprove the notion that organisms can arrive spontaneously from nothing. That is, take non-living material and an organism could pop up out of that inorganic non-living material. So for example, here I've got a bit of sterile broth. This was sterilized by putting it in an autoclave, which is a machine that goes to very high temperature and very high pressure, and it leaves nothing living in this broth. And so if spontaneous generation were actually allowed, then we would think that in this broth something living would pop up out of nowhere. However, it sits on the shelf and stays perfectly sterile. But it took scientists a long time to disprove the notion of spontaneous generation and to support or accept the idea of biogenesis. Biogenesis meaning that life comes from other living things of the same kind. Now, really, it should have been in the late 1600s when a man named Francisco Redi was doing his work that we should have just said no more of that idea of spontaneous generation. Francisco Redi was Italian, and this is a picture that I took last year when I took the ski team to Italy for the World University Games. We took them after the competition to Florence. Francisco Redi was a Florentinian. He was very much a scientist of the Renaissance. He was a fan, if we shall say, or maybe better to say a passionate lover of flies, and that is what he studied. But his contributions cannot be undervalued or understated, because what he was able to show was that life was not going to be present if the parent organism was not present to lay the eggs. And so let me show you what I mean. Now I've got a sort of derivation of Francisco Redi's study here, and you'll have to bear with me because I'm not using raw meat. I'm actually using a chunk of beef jerky in here. So in this jar is a chunk of beef jerky, but please bear with me and assume that if it were Francisco Redi, this would have been a piece of raw meat. And what he did was three things. One, he left the jar open to the atmosphere. Two, he covered the jar with a lid. Okay, so think of three separate experiments, one open to the atmosphere, one covered with a lid, and then finally there was a third one that was covered in some gauze. So there was gauze placed over the top of this jar. What Redi was able to observe was, in fact, that in the one that was open to the atmosphere, flies laid their eggs on the meat and maggots formed and the meat putrefied. In the one that was covered by the gauze, the eggs were on the gauze and not on the meat, and the meat did not putrefy. And the one that was covered, of course, had no maggots at all. The flies could not smell it, and they didn't even lay eggs on the lid. So this should have given us the notion that life comes from other life. However, of course, what Francisco Redi could not prevent was microbial life in here, and so there were still many proponents of the idea of spontaneous generation. Even when people showed that they could boil broth and that it would stay sterile in this bottle, there were still those who argued and said, yeah, but this is because air is needed for there to be spontaneous generation. That is, air must provide some epic, elusive, vital force that allows spontaneous generation to take place. And so this was a lose-lose situation because, of course, when air was present, so too were microorganisms. And so there was a lot of difficulty in disproving spontaneous generation. In fact, there was a researcher named Theodore Swan, and what he did was he actually heated up a tube headed into the bottle of sterile broth and showed that that wouldn't cause growth. But people said, oh, you must be destroying the elusive vital force. So it felt like a lose-lose situation. So it was very hard to disprove spontaneous generation. So onto the scene comes a man named Louis Pasteur. I bet you've heard of Louis Pasteur. He was an epic contributor to both biology, medicine, right, very much, and chemistry. And we're going to look at his ingenious experiment that really in many ways put the nail in the coffin of spontaneous generation. Here's what Louis Pasteur did, and it was multifold. Now I'm gonna write this down because one of the first things that Louis Pasteur did was that he actually passed air through a sterile cotton plug. So, one, Pasteur showed that microorganisms were present in air. This was a missing link that was needed. So we're just jotting down that one of the first things that Louis Pasteur did was to show that microorganisms are present in air. Forgive my S there, that is an S. So microorganisms are present in air, and he did that by sending air through a sterile cotton plug and noticing that there was indeed microbial life in the cotton plug. The second thing that he did was to show that air alone has no elusive vital force. Sorry about my terrible writing. It's hard to write on this pad, and I haven't had practice in a while. So air alone, that's an A, has no elusive vital force. Now let's talk about how he went about showing these two things. It was to build something called a swan-necked flask, and you can see here that this has a neck on it that very much has a local minimum that allows the dust to settle out. So in this first bend of the flask, the dust and microorganisms carried in that dust settled out. The air continued to flow into the boil broth. Now the boil broth, Louis Pasteur was able to show that it stayed sterile for years, despite the fact that it was readily exposed to oxygen. So there was no elusive vital force in the air that would allow for that growth to take place. So this was the epic experiment that really disproved spontaneous generation by trapping the microbes in the neck, allowing air to access the broth, but yet growth never occurred. In fact, it didn't grow unless Louis Pasteur broke the flask neck and allowed exposure to air that hadn't settled the dust out of it. So this was a really phenomenal contribution to biogenesis. However, many of you out there know that the number one thing about doing science is that science has to be able to be repeated. So this is something that many scientists struggled to do after the work of Louis Pasteur. So while his research really did disprove spontaneous generation, the lack of ability to repeat his work was troubling. And so there were some additional scientists that had to come onto the scene to help further support Louis Pasteur's work. John Tyndall was very much fascinated by dust, and in fact I have a very interesting address that he gave. It's written in the British Medical Journal, date 1871, where he's talking about the link between dust and disease. And I want to read a passage from this to you because it is incredibly sexy. You know that as regards the lowest forms of life, the world is divided, and has for a long time been divided into two parties. The one affirming that you have only to submit absolutely dead matter to certain physical conditions to evolve from it living things. He's describing spontaneous generation there. Isn't it crazy to think that the world was divided into two parties that were very, very much at odds when it came to whether life came from other life? It's crazy to think about that and to maybe apply it to some of the things on which we are currently divided. The others, without wishing to set bounds to the power of matter, I love that, affirming that in our day no life has ever been found to arise independently of pre-existing life. Many of you are aware that I belong to the party which claims life as a derivative of life. So he's claiming himself as believing in biogenesis. So what he was able to contribute, of course, was this idea that microorganisms are found in dust. Of course, we know Louis Pasteur further contributed in that area. And he was further able to show why Louis Pasteur's experiments were not being accurately repeated, and that was because of the different kinds of broth that people were using. So Louis Pasteur had used beef broth. However, what other scientists found is if they used broth made from hay, they could not repeat his work. John Tyndall was able to show that different kinds of broth had to be sterilized for different amounts of time in order to enable the repeatability of the experiment. This, of course, gave rise to the idea that there must be multiple forms of life, that is, some that are resistant to heat and others that are not. And very shortly that same year, Ferdinand Cohn was able to show the existence of what we now know as endospores. Endospores are the resistant, dormant structures that form when the going gets tough. So when the going gets tough, the tough form endospores. And these resistant, dormant structures are in fact, incredibly famous, even for those of you in your lifetime. Many of you remember in 2001, the bioterrorist scares, when highly purified endospores were sent around in the mail. These were endospores of Bacillus anthracis that people were inhaling and getting pulmonary anthrax from them. So endospores, even a century later, are still very much nebulous to us, in that we had trouble treating many of those cases. So we can look at a timeline and answer our original question. That is to note that even after the work of Antony van Leeuwenhoek, it took over a century before we started to see the contributions that would disprove spontaneous generation. So the answer to our question then is 139 years ago. That's all it was that we finally understood life as coming from other life.